Hello everyone, welcome to Easy Biology. The topic is about principles of inheritance and evolution. So in this we are going to discuss about test cross. So what is test cross? See, for example, in F2 generation or F1 generation, whatever the hybrid obtained in this generation, we cannot know whether the genotype is whether like this, either like this or like this. Okay, so whatever the phenotype is there, it is not possible to determine the genotype by seeing the phenotype of a dominant trait. Phenotype. Okay, for this purpose, to know about the trait at F2, Mendel crossed. Okay, so Mendel crossed F1 hybrid with Mendel crossed F1 hybrid. So to determine the genotype of a phenotype of dominant trait, to determine the genotype. Why? Because if the phenotype is tall, we cannot able to know it is tall because of this type of genotype or this type of genotype. That is why to determine it, Mendel crossed F1 hybrid with recessive parent or dwarf, and Mendel called this as test cross. Next, so how he did this one is, for example, this is a flower and whose color is unknown, means the color is dominant, flower color, but whether it is tall or whether it is means flower color, whether it is violet or it is violet because of this one. Okay, this flower is either contain this type of alleles or this type of alleles, means genotype. Okay, why? Because either like this it is violet color, either like this it is violet color only. So for this purpose, what the Mendel did is, here he taken recessive character, homozygous, means small w, and here capital W. Then what about the obtained all are, the phenotype obtained all are violet color, whereas here capital W, small w, here recessive, here also recessive. Why? Because here what we are doing, we are crossing the F1 hybrid with recessive dwarf. That is why we are taking the alleles here like this, and here small w. So here capital W, small w. Next capital W, small w. Okay, means here this W, small w. Why this situation occurs means, see here this is recessive and this is heterozygous. Okay, so if we observe here in this case all are violet, whereas here and all are violet and homozygous, whereas here heterozygote is formed, and half are violet and half are white. Half are violet and half are white. Okay, so general definition for test cross is the crossing of F1 hybrid, crossing of F1 hybrid with or heterozygote, crossing of F1 hybrid or heterozygote with dominant phenotype, then no recessive offsprings are produced. Okay, then no recessive offsprings. The crossing between F1 hybrid with dominant phenotype, then no recessive offsprings are generated. Okay, this is called test cross. And what is back cross? Okay, so test cross is a kind of back cross. Test cross is a kind of back cross where in this the crossing of F1 hybrid or heterozygote with any one of the parent, and they are phenotypically and genotypically similar with parent. Phenotypically and genotypically similar with parent. Okay, is called back cross. Okay, so by observing all these observations, Mendel proposed a law. Okay, he proposed two laws. One is the law of dominance. What is law of dominance? Is one allele is dominant over another allele. This is called law of dominance. So in this dominance, what he said is always the alleles appear in pairs. Okay, always the alleles appear in pairs. And who are responsible for expressing a trait is factors. Okay, for expressing a character, who are responsible is factors, and these factors are in the form of pairs. If the pair is dissimilar, then one of the allele from pair is dominant, and one is dominant and other is recessive. So this is called law of dominance. Next one, law of segregation, or this is also known as law of purity of gametes, law of segregation or law of purity of gametes. So what is this one is, Mendel stated that the separation of alleles during gamete formation is called law of segregation. The separation of alleles during gamete formation. And how he explained is, for example, this is the red color flower and this is a white color flower. He cross pollinated both, then obtained all red color flowers. Okay, now again he self pollinated this one. Okay, then capital R, small r, smaller. See, these are red color. Capital R is red, and capital R, small r is pink. and small r is white. So here phenotypic ratio is 1:2:1 and genotypic ratio is also 1:2:1. Okay, phenotypic ratio is 1:2:1, genotypic ratio is 1:2:1. So what the Mendel is saying is, for a pair of alleles, only one of the allele from the pair only during meiosis or during the formation of gametes passing to the zygote from parent. So while formation of gametes or during meiosis, only one allele from each pair passing to the gametes. This is the distinct and this is the common thing that is going to happen naturally in sexually producing organisms. Okay, this is about the law of segregation.